appropriate study design for clinical studies. When we talk clinical studies, it's basically dealing with the humans. So, what we want to address basically in analytical epidemiology, whether E to D or D to E. Basically, E is our exposure and the D is the disease, whether they are linked or the outcome is related to the exposure. A study design is a specific plan or protocol for conducting the study which allows the investigator to translate the conceptual hypothesis into an operational one. So I come into the term called hypothesis. So this clinical study involves research using human volunteers and participants that is intended to add medical knowledge. And there are two main types of clinical studies. One is the clinical trial or independent studies and observational studies that we already talked about. A good clinical study starts with a good research question built on a good hypothesis which is based on well-constructed comprehensive view. In to this context, I should say that the good science is good ethics, and all the research has to go through the ethical norms. And one of the major point in ethics is a good clinical study, good protocol. So when a study design sequence, when we talk about, let's think about, say, we want to formulate one hypothesis. We had some reporting of cases, which called case report. A number of case reports come to case series, which constructs our descriptive epidemiology. From there, we come to analytical epidemiology and animal studies and laboratory studies. Analytical epidemiology again built out hypothesis testing, the clinical trials, then hypothesis testing studies are cohort, case control, and cross-sectional, which has already been talked about. So now this type of clinical studies and designing the design. So whether these exposure we assign, exposure or intervention we assign or not. If we do not, if it occurs naturally, this intervention or the exposure, it is observational study. If we assign the intervention, it becomes an experimental study. So now these assigning the intervention, whether it is random or random allocation or not. If we randomly assign, it is randomized control trial. If it is not, then it becomes a non-randomized trial. Again, in the observational study, if we have a comparison, a comparator, in this regard, there is one good saying that I should say, mention about George Bernard Shaw. When she was asked how his wife looks like, she said, compared to whom? So we need a comparator. If it is not, then it becomes just a descriptive study. If we have a comparator, then it becomes analytical study and the direction of exposure. Now, it depends on the direction of exposure. In a cohort study, we come from exposure to the outcome. And the case control study, as was discussed earlier, from outcome to the exposure. And in the cross-section of the snapshot studies, exposure and outcome at the same time. And the cross-sectional study can be many times, snapshot can be taken many times in the forwarding time, which we call this longitudinal cross-sectional study. Thus, step 1 and step 2 is thus defined whether the study has a treatment or intervention, or whether the study is observational, usual in the clinical practice. And in the step 2, that is very important, whether we assign or not. If we do not assign, it becomes an observational study. And if we assign, it becomes an experimental study. For experimental studies, distinguish whether the intervention will be assigned by truly random technique, then it is an RCT. If it is not, it is a non-randomized thing. So this is an experimental study design that we can think of like random. This is a study population or the study subjects or something. We randomize. One group receives the intervention and one group do not, with all of the standard care of treatment, which becomes the control. So there is an outcome. There can be two possibilities, dichotomous. There is an outcome, there is no outcome in both the groups. And again, at the baseline, it is a forward study. And we measure the outcome in future. It becomes a forward study or a prospective study. Basically, experimental study design or RCT or a randomized control trial is a variant of cohort studies that has already been talked about. We forwardly we look into the time. Just so just so the difference between the experimental studies and the observational studies is basically uh, it occurs in a controlled environment, the experimental studies. But non-experimental studies is non-experimental, it happens. And it's a planned, we have a planned research designs, observational because there is no individual intervention, observation. Clinical trials are mostly well-known experimental designs. Clinical trials use randomly assigned data. We randomly assign and it gets the data. And in the observational studies, treatment and exposure occur in a non-controlled environment. And community trials use non-random data in experimental studies, but in observational study, individuals can be observed prospectively, retrospectively, or as in cross-sectional study occurring currently. So now let's talk about the randomized control trial. Only randomized control trials of sufficient size can adequately control for known and unknown confounding variables to minimize bias. Two things that have come up. One is the confounder, one is the bias. So this is a big thing. When we talk about confounders, we have got affiliation to both the exposure and the outcome. And bias means it could be differential or it could be non-differential. Like no substantive difference be occur between group except study intervention, which is randomly assigned. Difference between groups in predefined outcome can be attributed to the inversion being studied. So RCT, the first step is begin with starts with the valid question. So and it should be clinically relevant. Greatest impact if limited information or high variability in care or outcomes. And it can be answered by properly designed an RCT. And whether it is feasible to perform at your center or not is a specialized thing. So now, if you look at the observational versus randomized trial, what happens? This distribution of baseline factors that may become the outcome, the age, medications, comorbidities varies in study groups. Known, these are known factors or known confounders which can impact the outcome. These are collected. And statistical adjustment we do matching. For known unknowns, known to impact outcome, it cannot be collected. Unknown unknowns, we do not know whether they either impact the outcome or not, and they were not collected. The randomized trials, all factors, known and unknown, that may impact outcome equally distributed among study groups. This is very important. The importance of randomization that it can control the unknown factors also. So why go through experimental RCT? If we look at we want to collect the evidences to test our hypothesis. We want to prove or disprove the hypothesis. So it is the highest level of any study design till now. If we start with the expert opinion, background information, there are many things like that. Expert opinion or background opinion, these are very promising. And the supported evidence are case report, case control study, cohort study, RCTs are your supported document. And this improves with the quality. But the best one is the multiple data, multiple study sources, which causes constitute our meta-analysis and systematic review, which are well supported and is the highest level of evidences. RCTs are basically structured comparative control experiments in which two or more interventions are studied across groups of individuals who are randomly assigned to receive the interventions. The randomization reduces selection bias and as well as judgmental errors. This is very important in the sense that in all the researchers, when we develop a hypothesis, it becomes our we love the hypothesis. We want to prove. Basically, instead of disproving it, we want to prove that our intervention or our study or my technique or my drug is better than what is currently available or what is not. So there occurs some kind of at the back of our mind, when we select our study population, we select the patient, there occurs some bias. So randomization tries to nullify that. So another important thing, and I said the two concepts, one is the finer, another is the picot. What is finer? It should be feasible. All aspects of study able to be completed. It should be interesting, prominent issue without a solution. It should be novel, fills up gap in current knowledge. And how this is avoiding duplication. There is a tool that has come up that I will discuss. It should be ethical, protect right and welfare of the patient, which is very important, and it should be relevant. And in terms of PCOT, what is that? First of all, we need to define the population. Say healthy Indian infants. I'm talking about, we recently concluded the one study which is published in a big journal, that you want to see the safety and immunogenicity of Rotavac and Rotacil. These are the two rotavirus vaccines currently available in the Indian public health scenario, administered in an interchangeable dosing schedule. Like it gets, there are 3 doses of one vaccine and 3 doses of other vaccines. We, in between the doses, we interchange and want to see whether the comparator with the normal dosing, the interchangeable dosing is equal or not inferior to the normal dosing. So with that, among whom we did the study, P is our population, the healthy Indian infants. Then I is the intervention, that is the interchangeable dosing regimen or like of Rotavac and Rotacil. C is the comparator, single vaccine regime, that is Rotavac, 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 or Rotacil, Rotacil, Rotacil. O is the outcome. What is our outcome? We showed the safety and immunogenicity of the mixed vaccine regime. Time frame is the timeline. Here is not applicable. For how long we want to see measure the outcome. That is our time. So now again, there's another. I want to show there's another example. We want to see whether the currently available oral cholera vaccine, it is given 2 weeks apart. 2 doses given 2 weeks apart. Now want to compare that with the 4 weeks apart, when it is given 4 weeks apart. So because 4 weeks, that is a technical thing. Why 4 weeks? Because it will increase the immune response. That is our hypothesis. Is an which is better in an endemic setting or not. We want to measure if we increase the dosing interval, whether it is more immunogenic or not. That is our research question. Here, if we describe the PCOT, P is the cholera endemic setting of Kolkata, the population, the adult population. I is the extended 4-week dosing schedule for OCV. C is the conventional 2-week dosing schedule of OCV. And O is the outcome, which is the immunogenicity, non-inferiority of safety. There's another term that I just put in, non-inferiority, we will discuss later. And T is the duration of the study data collection. So now what is the favoring randomized in trial? Why? Why do we randomize? This is, as I told you, ensuring unpredictable assignment, or there is unknown confounders. See, if we, on my left, it is the number of study population subjects or number of study population, to avoid any selection bias at this stage of a planning a study protocol. Randomization ensures no rule or predictability for allocating subjects to treatment or control groups. There are various methods of randomization. We can do simple, I'm coming to that, I will discuss that later, how we randomize to control the unknown confounders. And it promotes the comparability. Uniform distribution of known and unknown confounders among treatment and control groups allows for statistical inferences on the treatment effects, ensures that the other factors except treatment do not affect the outcome. We try to do that. There may be unknown confounders which creeps in. That is why the concept of confidence interval or the precision comes in. Because if we say, discussed earlier, that is what is 95% confidence interval for the precision. Why the precision or confidence interval sets in? That there may be there may be some creeping of error may come in, but their error is as extreme as higher is the precision. Narrower the precision, the unknown error is less. This enables investigator to control for any changes in the instrumentation, delivery of intervention, etc., since changes or drifts in measurement should affect both groups equally. If there's any question, please you can put in the comment box or you can ask me now. So these are the big things, but I'm just touching upon. Uh, so now designing an RCT randomization allocation of consignment and uh, what is that? Allocation consignment is the process. This is very important. When we allocate a treatment or intervention, it should be concealed. Is the process that prevents any trial participant or investigator from knowing in advance the treatment to which the subject will be assigned, so that the bias do not creep in. So, if the trial participant knows that I am getting the treatment, or I got the active intervention, or say a vaccine, so I may falsify, I may falsely get my attitude may change, and my behavior, my way of living may change. And if investigator knows that the participant gets the active one, so I'll take care of them better. So concealment of allocation is very important. It is different from blinding. So when I'll talk about blinding, this will come. There's another important thing. It is important that the decision to enroll a participant is made in uh, in arrogance of the treatment to which they will be assigned, as this knowledge may influence the decision on whether or not to enroll. There is another thing, that if I find the subject is very, sick, it may have an adverse outcome. I will not, I will assign him the safest, the control. I will keep him in the control. There is also a bias. So allocation should not be confused with blinding. This is very important. Allocation consignment seeks to prevent selection bias. As I told you, subjects with different characteristics being entered into a different group, versus blinding seeks to prevent performance and ascertainment bias, like different response rate to treatment. If he does not get well, so I am not sure whether he gets vaccine, whether the active one or not. So that is one part. Or to measuring effect of treatment due to knowledge of which the treatment was received. Is it clear? Concealment of allocation versus blinding. So when we do allocation concealment, before randomization we do allocation concealment, and after randomization we do the blinding. This is the method. It is called SNOSE. It should be sequentially numbered, opaque, sealed envelope, or pharmacy controlled, numbered, or coded containers. These are the methods of allocation of concealment. We usually keep it in a sequential numbered, opaque, sealed envelopes, and it stays with person or in an area which is not accessed by the investigators. And this pharmacy control, that the pharmacy people will control numbers and coded containers who are not part of the trial. And another way of concealment is central randomization. In a multi-centric study, it happens that by telephone to a trial office, they just call them and this is the number. On oral method, whose description contains elements convincing of concealment. A secure computer assisted method. Nowadays, it is always a computer assisted method of, nowadays we do not do manual randomization. Concealment allocation is much easier. It is done through a computer assisted method. So there's another study method that is called pre-test and post-test with control group design. What is that? That is, we randomly assign to the experiment group the treatment in the experiment group or the control group. In the pre-test or O1, if the pre-test, both the groups, O1 is the baseline one. And we provide the experimental treatment, which is X, and we get a result. And in the other arm, we do not provide the experimental treatment, and we get another result. And this, we compare these two results, O2 in the experimental treatment versus the post in the non-intervention arm. So that is called the experimental treatment to those in the experimental group. Pre-test and post-test are those measurements of the dependent variables that are made before and after the experimental treatment is performed. All true experimental designs have subjects randomly assigned groups, have an experimental treatment introduced to some of the subjects and the effect of the treatment observed. It may so happen that the effect do not occur in all the experimental subjects. It may occur some. So we compare between this. So there is another one is the after and the post test only control group. Here we here hello. Can you hear? This is also controlled the only control group. This is composed of two randomly assigned groups, but neither of which is pre-tested. We do not pre-test. Here, if we look at this one, we pre-test and we compare with the post-test. Here, we do not have the pre-test data with us, only the post-test. In this, like the critical care unit noise and rapid eye movement, REM sleep study. In this example, the authors, what they have shown, recruited participants with no hearing or sleep problem to determine if critical care sounds levels suppress a rapid eye movement slope. So no baseline or pretest is required. We chose the patients with those who have no hearing or sleep problem. We did not measure whether they have really sleep problem or not. So this is another method. Another design is basically factorial design. This is complicated, but this is very interesting. Here, two or more intervention comparisons are carried out simultaneously. Now, what happens? Thus, for example, participants may be randomized to receive aspirin or placebo and also randomized to receive carotene. So let's see how it is done. So hypothesis one is basically a single 325 milligram aspirin tablet taken every other day, alternate day, would reduce total all-cause mortality by 10%. That was our hypothesis. And more specifically, cardiovascular mortality by 20%. That is the all-cause is 10% and say total cardiovascular mortality is 20%. This is our say hypothesis one. And hypothesis two is beta carotene reduces the incidence of cancer. We want to study both. How we do it. So first of all, they should be consenting and something. So and one group, we put 25 milligram of aspirin. In another group, we placebo or inert, which is a control arm. There is 1:1 allocation. Among this active aspirin, we give that beta carotene, and another group we do not give beta carotene. Again, in the aspirin placebo, we give active carotene and we give carotene placebo. The second factor in a 2:2 design should not complicate the mechanics of the trial operation. That is very important. And maternally affected eligible requirements or side cause effects which might lead to poor compliance or dropouts. In physician heart study, beta carotene meets all these requirements. So here, here if you see, we provided two intervention in a single group of patients, and we measured two outcomes in a single setting. And now, what is crossover design? There is another thing called crossover design. That is the population put under intervention study, randomly allocated to intervention. Another is a control intervention, and after a washout period, the intervention group becomes the control intervention, and the other groups become the control group becomes the intervention study, and we do take the result and we study. So it yields a more efficient comparison of treatment than a parallel group. Fewer patients are required, might be required in the crossover design in order, because the same group once becomes the case, one becomes the control. In ideal situation, exposure in a case control study also, the exposure level should be similar. That is very important. In ideal situation, case crossover design, we do it sometimes. Towards each participant with two or more sequential treatment in random order, usually separated by a washout period. Washout period means the effect effect of the intervention goes out. After that, we do that. So in this type of trials, we are not interested in whether there is a cure. There is a demonstration that a new formulation, for instance, a new generic drug results in the same concentration in the blood or not. So this is another way of measuring it. So example is our objective whether the diet intervention improves cardiovascular profile in patients with rheumatoid arthritis. So we have a specific diet whether the rheumatoid arthritis uh, gets better or not. Propose Mediterranean light anti-inflammatory diet improves the cardiovascular profile in weight stable patients with rheumatoid arthritis. So randomized, one get the intervention diet, other gets the normal or the control diet. But there will be dropout and other disease may not occur. And after that, they will be put on control diet. And the control people will be put on the intervention diet and then we measure what is the effect in that. And through the crossover design, we can ascertain the efficacy or effectiveness of the Mediterranean diet. But at the same time, we must remember this, though I say that the RCT randomized controlled trials are the best or the precise most evidence generating tool. But sometimes RCTs cannot be conducted. That we need to know which when it is not ethical, possible to assign intervention, like cigarette smoking and lung cancer. To study the effect of cigarette smoking, we cannot assign somebody to smoke cigarette for 10 years. That is unethical. H. pylori infection and ulcers. We cannot put H. pylori in the gastric mucosa to see whether they develop uh, uh, ulcers and cancer. Secondly, impractical large sample size, very low incidence, because in a Bayesian system, when we study, we do any work on Bayesian system, where the a priori incidence is important for sample size calculation. So rare side effect of medication, we cannot do RCT. Impractical long duration outcome requires many years to develop. Say development of cancer, 50, 40 pack years. We need to follow them for 40 years. Their RCT is not possible. And another example is a parachute example. This is very common. Everybody knows it that whether parachute is safe or not, whether parachute is efficacious or not in a plane crash. One group we give a parachute when we do not give parachute and ask them to jump. So this is not ethical. We cannot do that. It is certainly conceivable that people might investigate educational programs through RCT to encourage aviators to take a parachute with them. The ethical point here is nobody should involve other people in research that involves randomly allocating them to almost certain death. That is very important that RCT cannot be conducted. Another which we'd like to touch upon, which was is asked to, what is quasi-experimental? In experimental design, as we understood, the assignment of the intervention is randomly allocated by the investigator. Here, it is assigned, but not by the investigator, but the natural experiments. Participants are randomly allocated to intervention they receive, but are regarded as intervention, but as regarded as an intervention or control group participant by virtue of membership to a naturally occurring group, such as living in a particular geographical area or being treated by a particular healthcare practitioner. When do we do a quasi-experimental uh, design? There are certain situations when research methods using a random assignment would be unethical, such as providing public health care to one group while withholding it from another treatment group. When we know that that intervention or that treatment is good for the public health care, randomized control trial may not be best choice for researcher for practical reasons, such as the work involved in group design or cost of reading to a large sample size of participants without a particular attribution for data collection. Yes. Yes. So this is what are the advantages? Like uh, higher external validity is definitely than most true experiments. because is a natural, it occurs naturally. They offer real life world scenario instead of artificially controlled setting. Uh, and disadvantage is that lower internal validity is poor without randomization because it was not randomly assigned. Randomness is lost in quasi experiment, and higher internal validity than other non-experimental types of research because they allow you to better control for confounding variables than the type of study we do. And disadvantage that the use of retrospective data that has already been collected for other purposes can be inaccurate, incomplete, or difficult to access. And there may be other kinds of biases like recall and everything. So what is the difference? RCT is basically randomized to address confounding manually. Here, to address confounding, a priori matching, we usually do a priori or before we initiate the study, we do matching. Sometimes over matching or like matching reduces the cell size the number in each group, and that reduces the power also. And post hoc adjustment in statistical models we do. It is carried out in a controlled environment and quasi-experiment study carried out in a real program or field setting. It measures efficacy, it measures effectiveness, and it does not require program indicators in RCT, but in quasi-experimental, it generally use program indicators. So, example that I said that the investigate effectiveness of breastfeeding support program for mothers to explore the effectiveness of the pamphlet for mothers returning to work. So we want to see whether these are the educational intervention works or not. So why was this is a quasi typical this is a published study. Now this was published in to the 2021. Why it is a quasi experiment? Because these naturally occurring groups. There are two groups. The mothers already have come back to the work. The mother plan or some, and other there is another group planning to return to work. So intervention group women who plan to return to work, and the comparison group women who are already back at work at least 3 months. So they have already done their part. We did not do it. So if we look at this diagram, N is the groups, these are the observational measurement that we got. In X is the intervention in the groups which has already been naturally offered. Some those we did not ask them to come back to the work. They have come back on their own. And the other part is staying at the household. So this is a quasi experiment. So another part is the randomization. One is the simple randomization based on single sequence of random assignment. Simple randomization. This is the most common and simple by putting a coin, shuffle decks or cards, throwing a dice, yes or no. So you go here, you go there. The issue is that in large clinical research, simple randomization can be trusted. Because they made the chance of getting equally distributed between the intervention and non-intervention. But in a small sample size, this is a problem. Because they may be skewed. Then most of them can go to the intervention or may not go to the. There's a disparity between the groups. So that to avoid that, we usually use block randomization. That means equals so that the sample sizes in both the arms they are ensures balance. There is a balance, and the numbers remains the same. How we do it? The block size is basically determined by the researcher and should be a multiple of the number of groups, two treatment groups. We usually take 4, 6, or 8. Okay, example, two treatment A and B and block size of 2 into 2, 4. Possible treatment allocation can be A, B, 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 A, B, A, B, B, A, B, A, B, B, A, B, A, like that. So block randomization, another one is the permuted. We do not know whether there is a fixed block randomization, block size of say 2, 4, 6. Another one is the number is not certain. So to increase the unpredictability, so that we do not the investigator cannot make out which is the intervention, which is not. That is very important. The first patient is randomly assigned to one block, which dictates the group assignment for the next 3. This ensures the group size are just about equal at any time. This is a fixed block randomization. So issue is that there is a loss of consignment may be lost, meaning that investigators begin to predict which treatment is coming next. So, which may occur. I'm showing that how it happens. So, another one is stratified randomization. Stratified randomization is that addresses need to control and balance the influence of covariates. That is that, uh, say, among 5 to 15, we randomize. 15 years and above age certified. Uh, height according to length of the baby, height of the person, we do the stratification. So these covariates are controlled so that the covariates are equally distributed among among the different stratum. Specific covariate must be identified by the researchers who understand the potential influence, whether in a say for example, if age is a confounder, age can affect both the exposure and the outcome, so that age can be stratified. So complicated to implement if many covariates are to be complete. If there are one or two, this certification is good. But there are many confounders that to be certified. Again, I said that the in each cell the number becomes very less and it becomes underpowered. So then how to choose? How we choose randomization technique? So what is the sample size? If it is large, we can do simple randomization. If it is less than say 200, are the covariates that need to be controlled? If it is no, then we do block randomization. If it say it needs to be controlled so that they do not affect the outcome. So in that case, are the participants in the study continuously or all at the same time? If it is continuously, they keep on getting recruited. This is an important randomization, covariate adaptive randomization. Or if we recruit the subjects at the same time, we can do stratified randomization. Now, what is blinding? Blinding is a process in which one or more parties in a trial are kept unaware. So, both it may be single blinded, it may be double blinded, it may be triple blinded. Subject is single blinded, investigator is also blinded, subject is blinded, it becomes double blind. Monitoring committee, as the statistician, who is not part of the study but who is doing the analysis. He is also blinded along with the subject and the investigator. This is called blinded, so that no bias can creep in. This is a conscious or unconscious bias may creep in, so it cannot creep in. This is important because the bias can affect recruitment and allocation, care, attitude, assessment, etc. That I was talking earlier. It is also used to ensure the objectivity of the trial results. So, like how we do it. Identical appearing therapies, real versus sham surgery or procedure is very difficult though. Surgical team uninvolved in further care or assessment because after the surgery, the assessor, who has done it or the assessor are different. Double done. We sometimes do double done. Subjects receive identical active and control therapy together. Like, for example, when we give two dummy, say normal saline injection or two normal saline, two vaccines to both the groups to make it a double dummy. Side effect of a therapy may unblind the subject. That occurs, assess whether then occurs unblinded. Though we say it is blinded, but if we say study the impact of one antihypertensive versus a placebo, those who have got, the blood pressure comes down may understand that they are getting in the active group or not. So blinding sometimes fails. So these result in and this was discussed in details. I will not go into what are the important things that is we want to measure. There is a difference. That is one thing. So if you are sure whether the superior, whether there is a more or less equivalent or it is not inferior. So what is required is the precision, probability of finding difference which does not exist, which is basically type 1 error, and power 1 minus beta, the probability of finding difference when it does exist, we usually take 90% power, sometimes 80% power. So these are basically important things that we need to, and effect size. How much we want to start, how much difference that we want to make. So, and what are the trial stages? that already talked about, like earlier it was discussed that phase 1, phase 2, phase 3, and phase 4. Phase 1 is basically your proof of concept, or we usually do it 20 to 30 to study the safety. In all the phases of study, please remember safety, 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 safety is the most important thing. In ethics, in Belmont report, which is following International Conference on Harmonization, it says that do not harm. We cannot harm any subject or any participant, whether my impact is whether my intervention is better or not. That is so phase 1, phase 2, phase 3, phase 4, safety is very important. So you usually evaluate safety, safe dosing schedule, identify side effects. In phase 2, basically effectiveness, further evaluate safety. Phase 3 is basically divided into 3A and 3B, efficacy and effectiveness. Phase 4 is usually provide additional information after approval, risk, benefits, and uses, like the post-marketing service, PMS. Post-marketing surveillance, where it is in the market, it has been given to millions of population and to now we want to see how many side effects we get. So that is the PMS or post-marketing surveillance. So these are the basically four types of trial stages, and it has got sample size issue is also there, but value assumptions are basically the similar. So what are the non-clinical study basically, which is done in vivo in laboratory animals, pharmacokinetics, toxicology study, and efficacy of the intervention. So it could be a field trial, an experiment with subjects not defined the presence of disease. Main focus is initial occurrence, need a large number of subjects. Subjects are not active healthcare, require visiting them at work, home, school, or other designated centers. Expensive and resource intensive, and these are very expensive. A large sample size. We should study prevention of either extremely common or extremely serious disease. Basically, vaccine trials are more or less field trials. Now it's to be we do the immunogenicity when correlative protection can act as a efficacy. Now, vitamin C in prevention of common cold or paralytic poliomyelitis is basically a field trial. Community intervention trial is extensive, cost-effective. An example, water fluorid is ecological study that we do, health education program. So cluster randomized trial, when unit of randomization is a group of household, group of population, which are called clusters. And disadvantage is complexity in design and statistical analysis. Example, vaccine coverage of a region. So this is a typical example. We want to study the effectiveness of VI polysaccharide vaccines. So objective is to use the protective impact of VI polysaccharide vaccine when administered under realistic public health condition. So I will show you just the, so these are the 80 clusters, 40 clusters received the vaccine under consideration at the VI typhoid, 40 received the hepatitis A. It was a coded. So this is the analytical strategy. So in the cluster design, we can get the indirect protection or hard protection also. So total vaccine protection is a comparison of typhoid fever rates between A and C. This is a total vaccine protection. Indirect is with a comparison of typhoid fever rates between B and D. B those in the non-recipient, and D is the non-recipient is the control cluster. Overall is between A plus B versus C plus D. So in a class, only in cluster design, we can get indirect protection. So population for analysis is two types: intention to treat. All randomized patients are included in the final data set. Per protocol is only patients who complete the trial according to protocol are analyzed. There may be question of loss to follow-up. Those who are lost to follow-up, say after a certain period of time, are not included, or just taken only that percentile contribution is taken into account. So intention to treat, as I explained, all patients randomized regardless of follow-up is there. Per protocol, they have to complete the protocol. They have to complete the follow-up. Did not receive sufficient study intervention were excluded. Did not return for adequate follow-up, and all excluded. So this one is the basic principle. I'm not going into details. The slide will remain with you, and you may record it. you may get, where we do usually strength in part protocol proof of therapeutic concept can be ascertained, but in part protocol analysis. And in intention to treat is randomized and protected from bias, and this is effectiveness, and it happens in real life. Intention to treat is very real life scenario where there is always a chance of loss to follow up. So complete follow-up or patients is a very important thing in RCT. It's another requirement, high methodological quality. And if numerous patients are lost to follow-up, results of the trial may not be accurate. Predefined method to deal with such patients. Last observation carried forward. Imputation method. We can use imputation method sometimes if there is a loss to follow-up. And recalculate results assuming that patient lost to follow-up in new treatment group had bad outcome and patient lost to follow up in control group had good outcome. That is, this is a very conservative approach we sometimes take into account. And recruitment and retention is an art in a clinical trial because it's a forward study, prospective study, cohort study. In any cohort study, we must remember that this to prevent the retention is an important part. Like engagement, communication with participants before and throughout the trial, brochure, ads, social media, phone, text, emails, websites are very important. And benefits of participant for subject that has to be clearly explained. Benefits of participant research personnel with the academic, authorship, financial, that has to be clearly explained to the researchers so that they get motivated. And they identify, minimize barriers to participation. So that is comes from the experience. Easy access to study personnel and activities, participation as a non-onerous as much as possible. So regular follow-up is very essential so as to understand the outcome, interviews, examination, laboratory tests, adverse event detection, reporting, and for quality assurance, regular follow-up is absolutely mandatory. And we must have a contingency plan for retention and follow-up. Patient management, evaluation, data monitoring plan, protocol amendment or study termination, everything has to be ready in a clinical study or in a good research plan. Now, this is sometimes we know that efficacy and effectiveness. Now we know efficacy is done basically in a control situation, effectiveness is done in a real life situation. In effectiveness, we usually use intention to treat. In efficacy trial, we usually use part protocol analysis. So, as I told you, there is a sometimes we use superiority design, sometimes we equivalence design, sometimes we non-inferiority design. Superiority design is a we want to show that my treatment is better than the existing one because I am sure that is so it requires a greater sample size. But in non-inferiority trial, it is a subset of a superiority trial. I say that my intervention is not inferior to the existing one by a certain margin of inferiority, say 10%. If the current drug is effective or current vaccine is efficacious, let's say 70%, my vaccine is efficacious at least 60%, so that 10%. And equivalence trial, we where I'll show you when we use it. It is if my vaccine is 70% efficacious, the current one, my vaccine is either 60%, more or less 70%, not less 60% and 80%. It lies in between the equivalence margin. Where do we use superiority, non-inferiority, and equivalence? Nowadays, we use non-inferiority trial because as ethically we cannot use placebo right now. Example, superiority placebo control efficacy trial. Trials to show active treatment is superior to another one. Greater effect of lowering LDL cholesterol, better symptom control in asthma. So we use drug and we don't, what is available is better. Equivalence show two different formulation of the same active compound and equivalent pharmacokinetics required for approval of generic drug. There we use equivalence. And last but not the least, very important is now which has come up after 2009, that is registration of your trial. Because until and unless you register your trial with ctri.nic.in, so you cannot publish data through in 11 ICMG. The consortium has made it mandatory to register your trial prospectively before before you start your study, first enrollment of the patient or participant, you have to register your trial to avoid duplicacy, to avoid incompleteness. Previously, in 2007, when CTRI Clinical Trial Registration of India has come in, it was not mandatory in 2007, but after 2009, it was made mandatory. To prevent selection to publication and selection reporting of research outcome, it is always there, which is not favorable, not published, not known to people. Prevent unnecessary duplication. Help public know planned or ongoing trials into which they might get into enrolled. Give ethics review board consider approval of new studies of which similar work and data related. It is clinical trial. One is clinical.gov.in and CTRI.in. So in India, we have to register into CTRI.in. So one thing that there are 33 things that need to be filled in, like from the who is the sponsor, who is the investigator, what is the intervention, what is the full PICOT, what is when, if whether it's multicentric or not, whether it is TCGI cleared or not, everything has to be given. And date when the CTLI, when the date of enrollment, that has to be mentioned. So why register a trial? It is required by law now and enable publication, as I told you, and ethical. Register all intervention trial, your scientific, ethical, and moral responsibility. So clearances, the first two are basically say scientific advisory committee, institutional ethics committee, regulatory bodies. This is the Drug Control General of India. If it's an investigational new drug, IND, vaccine, like vaccine is always a new drug, which is IND, which there is a new formulation, new drug, or the change of dosing, it has to be clinical trial registry. Then if it, the funding you get from the external agency, your health ministry screening committee, sometimes you need to inform the state health, though it is not the clearance is mandatory, but you need to inform the local ministry, municipal corporation, human rights, police, so as to for retention as well as for preventing loss to follow. So now reporting, before I end, reporting a trial. When we do registering a trial, reporting a trial ensures all important elements of the study are included and reported appropriately through a structured method. It is called consort. Consort is framework for reporting RCTs required by many journals and checklists for content of the title, abstract, introduction, method, results, and discussion. And this is a consort for what is consort basically? This is a consolidated standard of reporting trials. It was developed from 1996 to improve quality of reports of trial. Now comprises 25 IPM checklists, and it was updated and revised in 2010. Extension added for cluster RCT, reporting of harms, non-inferiority trials have been herbal medicine, non-pharmacological intervention, as well as the abstract. So this is a consort 2010 flow diagram. So first of all, how many were assessed for eligibility, then screen failures, excluded, need not declined participation, other reasons, and how many after that, how many were randomized, allocated to intervention, no, not I mean control arm. And among those interventions, received allocation, did not receive allocation, intervention, give reasons why they did not receive intervention. Maybe the patient vomited, patient got sick and could not complete the intervention, so they went away. So that you need to mention. So loss to follow up, how many? And after that, your analysis comes, excluded from analysis. This is intention to part protocol. Again, on the control of all these things need to be given. So this is one typical example how we did it. This is a consort that we published in Lancet. This is more or less similar. If you look carefully, this is the how many I'm talking about the let's say the in placebo group 178 166 clusters were received the placebo vaccine 1727. So this is more or less comparable. So 32 individual lost to follow up. Here 37 individual lost to follow up, something like that. And what was the disease? How many discontinuous? How many incomplete? Everything has to be put in. Now we have need to put in the clinical trial, even the statistical analysis plan also should be developed and finalized in advance of breaking code, approved by the steering committee, data safety monitoring board, and regulatory agency. And good practice to conduct dummy analysis with arbitrary assignment of participant to intervention groups and who will freeze the data, your frozen data set. That is also important. Once the data is frozen, you cannot manipulate the data or change the data. That is that is very important, and we need to define everything that has been discussed earlier. What is the primary endpoint, exclusion and inclusion criteria, entry and exit, method of statistical assessment, any adjusted analysis or criteria for these, any subgroup analysis. So now the applying ethical principle. First of all, non-maleficence and beneficence. These are the three principles of Belmont report. Then principle of justice. Then principle of autonomy. If we call non-maleficence or beneficence, do not harm. We cannot cause any harm to any subject. Benefit maximum and risk minimization, reasonable risk and relation to benefit, and welfareness of participant take procedural of interest of science and society. So these non-maleficence, principle of justice, and principle of autonomy is the crux of any experimental design or any when we design a study, when we do an RCT, when we do even what experiment, we need to take care of that. And this has an immense importance in your translation also, because if it is not ethical, you cannot translate that knowledge into public health practice also. So just to summarize, base design, RCT or experimental design or RCT is the base design to minimize bias in assessment of intervention. Till now, factors to provide high methodological quality are like concept allocation, blinding, complete follow-up, predefined outcomes is there, sample size has to be taken proper care, analysis has to be predefined and pre-reported, pre-trial registration ensures no change of original design without explanation, reporting guidelines ensures all important elements provided in publication. Thank you very much.
Thank you, Dr.
So if there is any question or
question, would request all of them to type in the chat box or send it to Sumit, and then we will take care of that question in the interest of time. So thank you so much, Dr. Kamingo. Okay, first of all, thanks to all of you for listening patiently throughout a long day. It's such a long day, I understand. And keeping that in mind, we are postponing the last lecture to tomorrow morning. We will start at 10 o'clock tomorrow morning, just to long time. A big thank you to all our presenters for today. It has been a long day and they have done a great job. And I'm sure it has been beneficial to all of you. Let me emphasize that all of you are very good researchers. There is no second thought about your procedures towards applying to ICMR or the grant awarding body. But as Dr. Anita said in the morning, our DG just wanted to re-emphasize the fact that all proposals have to be very clear focus, and there should be a clarity of thought process in all the proposals. And with this end, that today's and tomorrow's workshop is planned. And thanks to Sumit for getting it through in such an elaborate way that we started with the research question, hypothesis, and research, the literature survey, and he has covered almost every aspect of methodology as well. And tomorrow we will be concluding by covering multiple aspects of data analysis, which is very important from the angle of reporting your results, both either as a report or as a manuscript. And then we will also be talking about certain additional aspects which are very critical from a discovery perspective, like your IPR and how to protect your data and those kind of studies. Finally, we will also be touching on the ethics and the, particularly from the social behavioral aspect, those things are also important. So most of today was mainly on the operational side of it, but tomorrow it will be more environmental, social, from a IPR perspective, and I do hope this and tomorrow I will it to be today. So thank you so much for being with us today, and I look forward to seeing you all tomorrow morning at 10 o'clock. At the same time. Thank you, everyone.